Welcome to the second day of the second Emirates Clinical Nutrition Conference. We will be starting this morning with our first session on advanced nutrition technology and genomics. Our moderator for the session will be Dr. Salah Ashikeli. Yes, Dr. Salah is the head of Nutrition and Dietetics Department in Sultan Qaboos University Hospital in Oman. He has a bachelor's in nutrition and food technology from the University of Jordan. He had joined SQUH in 1997 as a hematology and pediatric dietitian, bone marrow transplantation, BMT dietitian. He has been trained at the Great Ormond Street Hospital in London, UK, and has worked as an inborn errors of metabolism dietitian. In 2006, he has completed his MBA at the University of Hull in the United Kingdom in 2008, has completed the Masters of Nutrition and Dietetics in Griffith University, Queensland, Australia. He is also the member of the Dietitians Association of Australia, accredited practicing dietitian, and in 2014, he has joined the University of Leeds, UK as PhD researcher on the effects of dietary intake and gestational weight change at birth weight. Thank you, Dr. Salah. He will be leading to this morning's session.
Thank you very much for your introduction. Good morning, everyone. Sorry, how can I get the bio of the next speaker? Good, okay. It will be my pleasure to introduce Dr. Sundas Harfield. She's a lecturer at the Department of Medical Laboratory Science at the College of Health Science, University of Sharjah since 2008. She served in many academic institutes and presented in international and local conferences, holding a PhD in biochemistry from University Science Malaysia. She had a BSc and master's degree of biology from University of Jordan. She's interested in research. Research interest is focused on studying the gut microbiota by obesity and clinical intervention with probiotics. With no further ado, I would like to introduce Dr. Sundas, and the field is yours.
Assalamu alaikum. Good morning. It gives me a great pleasure to present today in the second Emirates Clinical Nutrition Conference for the second year. Thanks for the organizers for inviting me. I'm going to talk today about the gut microbiota and obesity and links with the host genetics. I'm specifically interested with the probiotics and studying the gut microbiota. But in today's presentation, I'm going to talk about the interaction with the host genetics and how the bacteria, the microbes living in the gut interact with the host genetics at the expression and the translational level. But basically, I'll go through the effect of the diet and other environmental factors that may contribute also to the changes in the host genes expression, as well as the alteration on the gut microbiome. There are so many factors that contribute to the gut microbiota, and the list is growing. So you understand there's a relation with the diet, the lifestyle, the level of the stress and the infant feeding method, the birth process, and even the geographical distribution. Population studies emphasize the effect of the population on the gut microbiota alterations. But first of all, I would like to differentiate between two terms, the microbiota and the microbiome. Microbiota is the community of all the microorganisms, the bacteria, the viruses, the fungi that coexist together in a particular habitat. And in our case, the guts. But the microbiome is the collection of all the genome of the microbes. And in any ecosystem, particularly the collection living in and on the human body. By numbers, we could say that the human body harbors more than 100 trillion of bacterial cells, and 90% of all the human cells are microbial cells. The microbial genes corresponds to more than 100 times the human genes. So we can imagine the effect of the microbes genes on the human genes expression. Looking at the individual, the human, more than 99% of the human genome is almost similar. So we have only differences in like 1%. But the case is not the same in the bacteria, and the microbial genome is very, very diversified. Less than 20% of the microbial genome is similar. So we can imagine the effect of the microbial genes on the human or the host genome in general. Of course, when we talk about the host genetics, we cannot underestimate the effect of the other environmental factors, the diet, the lifestyle, and the hormones as well, that also correlates with the microbial genes and the expression, as well as the effect on the host genetics. The microbiota composition in different organs varies. So we have different genera of bacteria that exist in the oral microbiota, in the gut, in the skin, respiratory tract. And as you can see, they vary at the genus level. So they are the names of the genus. So we can imagine what will be the diversity at the species and subspecies level. So it's a very, very diversified world. The intestinal microflora, starting from the stomach down to the small intestine and colon, it varies a lot, and the diversity numbers increase as long as you go deeper at the terminal end of the gastrointestinal tract. Obesity is not the only disorder which is only linked to gut microbiota. There are so many diseases, cardiovascular disorders, blood pressure, gut-brain axis, the autism, gut-lung axis, which affects the asthma, respiratory tract infection, and other gut skin disorders. So there are so many diseases linked to gut microbiota. But in today's presentation, I'm going to talk only about the obesity. And we all know that the microbiota should have, a kind of commensal relationship with the host and should play a key role in the human health and disease. So when this commensal homeostatic balance is disturbed, this will end up with diseases. Let's say, for example, the autoimmune diseases, allergic disorders, inflammatory disorders, and so on. But why obesity is, the scope of the studies worldwide is talking about the obesity and its relation with the gut microbiota. It's basically because of the alarming worldwide rates and the complex etiology of the obesity. There are so many underlying causes of obesity, like the unbalanced diet, the lifestyle, the genetic background, and the social environmental factors. Only in the early 21st century, it has been explored that there is a direct relationship between the diversity of the gut microbiota and the rates of obesity in different population studies. It's a very, very old experiment that proved when we use the germ-free mice. And in this experiment, the microbiota was transferred from the obese twins to germ-free mice. They developed obesity, while the microbiota transferred to germ-free mice from lean twins, the rats or the mice, they didn't develop obesity, although both groups of mice received the same diets. And definitely it has been proved that lean or normal weight people compared to obese or those who are diagnosed with metabolic syndrome, they have a great diversity in the gut microbiota, which affect the level of metabolic outcomes and the level of inflammation, plus and many other disorders. There's a growing evidence that supports that several microbial metabolites interact with the epigenetic process in a way that although we do have inherited chromosomes, inherited genes, but the level of expression, translation of these genes is affected by the microbial metabolites. On the other hand, it has been also under the discussion that even the human metabolites may also alter the microbial balance or composition. Any such kind of studies should be proved through microbiome genome-wide association. It's very difficult to reach to a conclusion from a pilot or small studies. It should be like genome-wide association studies on large populations over years. The different complex human diseases share standard and distinct gut microbiome features. It's better to understand the disease etiology by studying the effect of the gut microbiota in most of the diseases right now. The gut microbiota is mostly composed of 5 known phyla. I don't want to go through deep details of taxonomy of microbiology, but it's good to know that approximately 90% of the bacterial species belong to the phyla of firmicutes and bacteroids, and 10% for the other 3 different phyla. So any change in this, balanced distribution of genera will definitely reflect on the inflammatory disorders and the homeostatic balance between the host and the microbial normal flora. For example, the bacteroids enterotypes has been associated with diet rich in protein and animal fat, and the Prevotella enterotype was rated to high carbohydrate diet. So we can feel now that how the distribution of different types of microorganisms is linked to that type of diets. Dysbiosis, which is blamed to all the disturbance of the homeostatic balance, is defined as a disturbance to microbiota homeostasis caused by disruption of the ecological equilibrium in the gut and any change in the microbial gene richness, functional composition, as well as the metabolic activities. As long as the intestinal barrier cell membrane are intact, that means the bacteria, the microbes are living inside the intestine, and the metabolites are kept inside. But the exposure to high fats, high sugary diet may end up with some leaky gut or leakage in the cell in that intestinal barrier that leads to the escape of the microbial metabolites into the bloodstream. So the microbial metabolites are in the wrong place in the bloodstream, which will slightly elevate the level of inflammation and induce the metabolic disorders. Bacteroids and firmicutes phyla are dominant, and it has been found in dominant genetically obese mice. About 50% of them, they have fewer bacteroids than the other species, and correspondingly they have more firmicutes. That will approve the hypothesis of the elevated firmicutes corresponds to obesity. In a human, however, because the human, they are complex living organisms, it has not been confirmed yet whether the gut microbiota dysbiosis is a causal factor, but in the germ-free mice, it has been supported and proved that dysbiosis is the causal factor of obesity. And we have to put in mind that the human is exposed to different factors, environmental factors, social factors, but germ-free mice, they are living in controlled environments. So it's going to be somehow, it's a kind of a criticism that the animal models cannot be generalized on human studies. Some microbial species, especially firmicutes, they can undergo fermentation. So they are able to digest the undigested food and release short-chain fatty acid. That means they can elaborate or increase the energy expenditure. I don't want to go through these details because short-chain fatty acids is a long topic to talk about. Let me just take you now to the effect of the diet or the gut permeability. The high-fat diet contribute to the regulation of integrity of the intestinal mucosal barrier. So the high-fat diet will induce the dysbiosis. And the dysfunction in the gut permeability is a primary event triggering bacterial translocation, not only the metabolites to the bloodstream and the onset of the inflammation and metabolic disturbances. It has been also proven that the high consumption of artificial sweeteners may also disturb the gut microbiota. For instance, altered growth of gut bacteria was reported after administering the Splenda sweetener, and non-caloric artificial sweeteners as well, such as saccharin. It impairs with the glucose tolerance by modulating the composition of the gut bacteria. To transfer you now to the main topic of today's presentation, which is the genetic and epigenetic factors. There's a complex interaction between the host genetic background, gut microbiota, and the diets. Phenotypes and the gut microbiota composition among genetically distinct inbreed mouse strains in response to diet. Emerging evidence in humans revealed that expression of polymorphic genes related to obesity was conditional on exposure to the microbiota in primary human chronic epithelial cells. That means the level of gene expression is altered as a result of the change in the gut microbiota composition and their metabolites. In human, for example, studies, it has been found that there is a strong association between obesity-related genus, which is the Akkermansia. This genus of bacteria is correlated to a variant near the phospholipase D1, PLD1 gene, and this gene is associated with elevated BMI. So if you have elevated level of this genus, that means you will have activated gene, PLD1 gene will be expressed at higher level, and this will increase the basal metabolic index. Significant association has been also found between the abundance of the Prevotella genus and the human variant RS87A3 gene, which is linked to lysophospholipase like 1 gene in human, and this gene is strongly related to fat distribution and insulin sensitivity. So it also related to the diabetes. There are so many genes in the literature that I have tried to summarize, but it has been shown that epigenetic mechanisms are probably more relevant in early childhood. So such kind of changes may start at early time at the childhood and continues. The epigenetic could be related to gut microbiota colonization and development through that type of delivery, breastfeeding, and the introduction of solid food, infection, antibiotic treatment, and so on. Obese patients, for example, they have reduced microbial diversity. Well, it's very well known that obese patients, they have less diversified microbiota, and they have abundance of Faecalibacterium, which is, associated with the lower methylation level of the FFAR3 gene, which is correlated to higher BMI. There are also studies about the effect of probiotics to modulate the gut microbiota and its relation with the host genetics. Probiotics are potential modulators of obesity and its comorbidities, and they have well-known beneficial effects through many different ways. For example, probiotics, they may aid in stimulation of the beneficial bacteria and short-chain fatty acid production. Probiotics also may increase the level of some beneficial species like bifidobacteria that could contribute to restore the normal gut dysbiosis. And probiotics also, they can contribute to lipid metabolism modulation, possibly by inhibiting the lipogenic enzymes and decrease the synthesis of lipoproteins and triglycerides. Some probiotic species like Lactobacillus bifidobacterium have shown amelioration of obesity status and associated metabolic disorders in high-fat diets fed mice. And in our study that we have accomplished at the University of Sharjah, we have proven that some species of Lactobacillus, they have dramatically decreased the level of inflammation in human subjects. There are, lots of variations in the host genotypes that are indicated to play a role in microbiome assembly, which may explain to a certain extent the microbial stability and the resistance to alterations. Always when we look at the human, we have to consider that the human is a complex living organism which is exposed to a lot of environmental and social corresponding factors. The bidirectional interaction between host and genetics and the gut microbiome are recognized, exerting a profound influence on the host health. So, for example, the host gene AMI1, which encodes the salivary amylase enzyme, has been revealed to be associated with the host phenotype, such as BMI and metabolism. So those who have alterations, some species of microorganisms in the guts, they have shown some elevated level of gene expression. So some genes are overexpressed, over-translated in view of changes in the gut microbiome that will definitely reflect on the lipid metabolism and the BMI eventually. The interaction between the microbiome and the microbiome-derived molecules and hosts can occur in different mechanisms. We'll just briefly describe the events here in four mechanisms, A, B, C, and D. The first mechanism is the microbiome derived stimuli. That means the stimulation is coming from the microbiome. They are recognized by the host cells, and through this interaction, the stimuli include microbiome derived transcripts, small molecules, proteins and enzymes, or pH changes. So they affect the transcription level. Or could be through microbiome derived molecules that cause differential transcription factor binding, methylation, chromatin availability, so affect expression modification. Or it could be through microbiome derived molecules that can also lead to change in transcription and alternative splicing, resulting in different host gene products, so even chromosomal aberration. The last mechanism is the host-derived proteins. In this case, the host produce proteins due to gene expression, and these proteins, metabolites produced by the host, will change, affect the normal distribution of the microbiome. So either A, B, C methods that, they are microbiome driven, or D, which is host driven mechanism. Both ways. To conclude, the complexity of the interaction between the microbiome in the gut and the host is so complicated. Although recent studies in model organisms and the human demonstrate that trans-kingdom crosstalk occurs, there are still many avenues to explore. It's better to examine the influence of host genetics on the microbiome of the population in a controlled environment rather than taking the environmental factors in simple systematic analysis. That's it for today, and thanks for being with me.
Thank you, Dr. Sundut. It was a very heavy breakfast in terms of science. I hope everybody's microbiome is doing well today morning. So with no further ado, I would like to ask Ms. Farah hello to come to the podium. Farah has 15 years experience working in various fields, including clinical nutrition, corporate wellness, health promotion, and academic. Wow, she has very long bio. Mashallah. Okay, I'll just cut to the chest. I like this last sentence. The last sentence, Farah nutrition philosophy is to educate, empower, and that makes the most different and elevate health and promote evidence-based cutting-edge researches. So let us listen to Farah, and I hope that will be less. Oh my God. Thank you very much. Please welcome Farah with me.
Assalamu alaikum. Good morning. Yes, please embrace for an even heavier breakfast. It's two very complex topics, I think, for an early Sunday morning, but I think two extremely, extremely important topics as well. Um, okay. Why is this working? Oh, okay. Okay, so I think this is the vast majority of dietitians at any point during their career where they are bombarded with a variety of different questions and statements. Why am I not losing weight? Can I eat this? Do I need to avoid that? Do I need to take supplements? I'm always craving sugar. I'm always fatigued. And the dietitian or nutritionist, despite trying so many different techniques, is often feeling at a loss at any point during their career. So this is not unusual. So throughout my presentation, what I would really like to emphasize is despite so many tools in our toolbox where we do try to troubleshoot sometimes, there are certain questions that are left unanswered. And what I would like to do during my presentation is to give you that knowledge, that confidence that genes could really be at the core of some of the issues where we struggle to answer. So during the first part of my presentation, I'll go over really a background into genomics and then highlight the role of genomics, particularly in the area of nutrition, and then how we can integrate and embrace genomics into our practice. So this is a very brief timeline in terms of genes and genetics and genomics. So in the early 20th century, this is really where chromosomes we found out that we are made up of chromosomes, one coming from our mother and one coming from our father. And then around the 1910 to 1950, that's the discovery of the nucleotides. So adenine, guanine, cytosine, and thymine. And then we discovered that actually the A and throughout our genes, A and T are always bound together and the C and G are always bound together. And then throughout time, of course, the discovery that the DNA is in a form of a helix. In the the early 1980s, pioneering DNA sequencing methods, and the PCR strategy was developed. And then in the 1990s, there was an initiative to conduct the Human Genome Project. So this was launched and completed around 2003, actually. It was 2 years early. So the complete human genome was sequenced. And this really forms the base of where we are today in terms of better understanding and utilizing this information in order to conduct our research and better understand the makeup of a human genome. So in 2008, the 1000 Genome Project was launched, and this aimed to sequence the whole genome of a large population. And I will talk about population genomics later on. But first of all, let's go over a couple of definitions because there are quite a few terms that sound very similar yet have very different meanings. So the term genetics refers to the study of the genes and their roles in inheritance. So it's really identifying certain genes and linking those to certain diseases. Okay, so for example, the gene that encodes for phenylketonuria. Okay, so this is where, again, we understand what genes can lead to specific diseases. However, when we talk about genome, it's really the science of our genomes, okay, or genomics. So it describes a study of the interaction of our genes with the environment. Okay, so yes, there are specific genes that encode for specific diseases, but then there are other genes that can increase the risk of disease. And this, what determines whether or not they do eventually lead to disease, is really the interaction with the environment in terms of, let's say, diet, movement, stress, sleep, and overall lifestyle. And this is really what we tend to focus on. Well, we focus, of course, on all, but in the context of chronic disease and the many questions we have in terms of early prevention, detection, management of chronic diseases, it's really important to understand the certain genes that can predispose us to these chronic conditions. So nutrigenetics and nutrigenomics, again, they are often used interchangeably, but do have different meanings. So nutrigenetics is the effect of having a specific gene on nutrient, for example, metabolism or utilization. Okay, so again, it's what gene can impact what nutrient. So this, by understanding the makeup, a genetic makeup that an individual has, it can help us personalize their diet and nutrition further. On the other hand, nutrigenomics is the effect of our nutrition, of our diet on our genes. Okay, so it studies the effects of the variety, of course, of nutrients in our diet and the impact that they have on gene expression levels, and of course, eventually proteins and metabolites like, for example, hormones. So if we look over here again to continue talking about nutrigenomics, if you look, and this is this cascade, let's say, of the impact of nutrition on our overall health. So our nutrients again will impact our DNA or our epigenome, so the expression, let's say, of specific genes, which eventually will impact our transcriptome or our RNA in this, in, let's say, a specified genetic or cell population, which eventually, of course, will code for specific amino acids and proteins and metabolites. Okay, so any, let's say, if our nutrition, if you see there, if we rely on, let's say, a diet high in processed foods, then this is negatively going to impact our DNA, which will negatively impact RNA, and the protein that it encodes, and then the metabolite that it gives off, again, things like hormones, neurotransmitters, cell signaling, and this metabolite will eventually trigger things like inflammation, oxidative stress, insulin resistance, neurological stress, and then this really leads to disease. Okay, so ultimately, it's our nutrition, of course, as part of our lifestyle that can dictate that cascade and chain of events throughout our genome. So the traditional approach, this I'm sure is a figure that we've seen one too many times, and we probably use even in our day-to-day practice. Okay, now any thoughts about the plate? Do you like it? Do you not like it? Do you use it in your practice? Okay, تفضل. Yeah. Mhm. Yes. So it is a great tool, I think, for visualization. Did you have anything to add for that? Mhm. Yeah, so it's a great way to visualize and to really help explain. However, a lot of these recommendations, including, the way that plate was derived, are very general, right? And often, of course, with our clients, we do tend to personalize it based on their needs, okay? But in the era of personalized medicine and longevity and really optimizing health, it's so important to take maybe this as a foundation or base, but then to kind of revise it as per the individual's allergies, intolerances, culture, likes and dislikes. So it really is a once this it's we want to move away from a one size fits all approach, okay, and into more of a personalized approach. So we live in large modern populations with very diverse genetic backgrounds and ancestries that can have a range of metabolic responses to the same foods. Therefore, it's important to question, okay, yes, this has been used for so many years, but is it enough? So the human genome, and we are all, the vast majority of our genetics are actually very similar to one another. However, there is that 3% difference, but that 3% difference is really what can impact our overall health. Okay, so these differences, or what we call genomic variants, are what can increase or decrease our risk of disease. So if you see over there, let's say person one, the normal person, okay, sometimes just a single change or deletion or insertion of a nucleotide, the C over there became a T. For person number two, that variant can be what's going to predispose them to a specific, let's say, condition or trait, let's say insulin resistance, okay, or oxidative stress. Okay, so it's the simplest and most widely studied type of genetic variation is known as a SNP. So you might hear me talk about SNPs quite often. So there are over 300 million SNPs that have been found to date, and roughly 4 to 5 million genomic variants in an individual's genome. So we can have many of these variants. Some can actually be protective, and some can be more detrimental. Okay, so SNPs, again, these genetic variations will encode for a different amino acid, which will encode for a different protein, metabolite, etc. Okay, so that the differences in proteins, again, are what can be problematic. Okay, so the SNP can alter the synthesis and function of proteins, leading to changes in what nutrients we need and how we metabolize these nutrients. So per disease, okay, so in general, the large amount of genomic data that we have to date has helped us identify these SNPs and has helped us identify which SNPs are related to which conditions. And certain conditions can have, hundreds or even thousands of genetic variants or SNPs. However, we know that there are what we call monogenic diseases and polygenic diseases, and I think I touched upon this earlier. Monogenic diseases is where a single SNP or a single genetic mutation will lead to a specific disease. Again, things like sickle cell, phenylketonuria, cystic fibrosis. However, more polygenic genes, let's say, are basically, you could have, for example, cancer, cardiovascular disease, are related to many thousands, hundreds of thousands of different SNPs that are impacted by our environment and the choices that we make. Okay, so these are what we call polygenic diseases. They are caused by several or many different genes and defects in these genes. Sorry. We always say this in functional medicine, functional nutrition, where you can have a certain, let's say, a variety of different SNPs or genes, but ultimately, even if you have these genes, it does not mean that you are going to get that certain disease unless it's genes load the gun, whereas lifestyle pulls the trigger. This is also a very quick thing. So when we look into population genomics, and to understand a person's risk of developing a disease or condition based on their genetics, there is what we call a polygenic disease risk score. So it's really looking at a population, comparing the genes of people who have a disease versus a population who does not have the disease, and to take that one person and where do they fit in. So let's say if they are on the higher risk, the outliers, then they will tend to have a higher polygenic disease risk score. But it's important to understand that sometimes from different ethnic backgrounds, different countries, different regions across the world, these scores might change. So a lot of the data that we have is related to the European genome. However, I'll talk about the Emirati genome program shortly, and this is really where we are working very hard to obtain a genetic, let's say, understanding of our community and society. So this polygenic disease score, it's only a risk. It does not show if that person will develop, if that person will develop the disease or not. It's just a risk. Okay. Okay, I have quite a bit to go over. So this again just reiterates the importance and the interaction of the environment with our genes. And let's say if we have certain SNPs in oxidative stress markers, insulin resistance markers, methylation markers, if we don't provide the right nutrients and the right, let's say, lifestyle factors, that will eventually influence molecular phenotype and then the clinical phenotype. So these are just a few quick examples. As I was told, I have 5 minutes and I am still 40% through my presentation. Diets rich in, for example, processed grains, white rice, white bread, processed meats have been linked to or upregulate genes related to oxidative stress and cancer. Same with red meat intake has been linked to genes associated with colon cancer. A higher saturated fatty acid intake results in gene expression profile that is typical of glucose intolerance. Diets lacking in folate, choline are linked with dysregulation of lipid metabolism. Chromium deficiency, of course, and genes that are linked to chromium have been linked to insulin resistance. And then selenium, vitamin A, vitamin B12 deficiencies can all increase susceptibility to cardiovascular disease by upregulating pro-inflammatory genes. On the other hand, positive effects, so Mediterranean diet, monounsaturated fats, particularly olive oil, and then bioactive compounds, sulforaphane in broccoli, curcumin in turmeric, EGCG found in green tea, can all upregulate, anti-inflammatory genes, and they can downregulate genes, let's say, that code for cancer and cardiovascular disease. And this is also just to quickly show the different potential genetic SNPs that can occur in the liver, phase 1 and phase 2 detoxification. So if you look into the yellow box, it shows that these are the cofactors, the nutrition cofactors that are needed to promote the function of these genes, which are crucial in terms of liver function and detoxification. And again, the variety of different genes and the polymorphism and the impact that they have on other conditions. So this is where, again, I went over just a bit of background in terms of genomics, what genomics is, defining SNPs and genetic variants, how we are implementing this in our practice, and then, how can we continue to integrate this? So we look into there's population genomics, clinical genomics, and then consumer genomics. Population genomics is really helping us understand at a larger scale all the genes or genetic variants that are more inherent in a particular population. And this is where, no doubt you've heard of the Emirati Genome Project or program, which aims to explore genetic makeup of Emiratis using cutting-edge DNA sequencing and artificial intelligence technologies to generate high-quality, comprehensive genomic data about the Emirati population. And this will really help not only health professionals, but public health authorities plan for, let's say, public health initiatives to really help cutting healthcare costs and helping healthcare professionals like us better address chronic diseases through dietary and lifestyle management. In clinical genomics, and this is also applicable to consumer genomics, it can help develop improved diagnostics, more effective therapies, provide evidence-based approaches for demonstrating clinical efficacy, and provide better decision-making tools for patients and providers. So it can really help at each stage of from disease prevention, screening, management, and even treatment. Consumer genomics, and again, this is applicable to nutritionists. This is where there has been a boom over the past couple of years in terms of, test kits where you either use a buccal swab or a saliva test, and then you wait for 3 or 4 weeks to get your genetic profile. And based on your genetic profile, what changes you need to make, let's say, to your diet and lifestyle. So in this case, let's say someone has had a red flag in terms of weak vitamin D processing, then in this case, how are you going to guide them? Okay, let's test your vitamin D levels. And then based on that, do they need certain supplements? Could you increase some of the, small amounts of food that could potentially contain vitamin D, and then of course promote, exposure to sunlight? Could they have maybe a sensitivity to gluten? In this case, you might do some further tests, maybe an elimination diet to better assess if this person, does need to be on a gluten-free diet or not. And this is more of the plate that we should be focused day, as opposed to the one that we tend to focus on, which is the other one. So really, it should be very personalized, based on dietary habits, of course, food behaviors, physical activity patterns, deep phenotyping, our microbiota, as Dr. Sundus, so very well put it, nutrigenomics and metabolomics. So it's precision medicine. This is what I was just talking about, direct-to-consumer genetic testing. Again, there are many ancestry-related ones, but then there are ones, 5 more minutes, 2 more minutes, that are related to genes. Again, they can look at hundreds and hundreds of your genetic predisposition, whether it's cancer, cardiovascular disease, food allergies, food intolerances, the way you metabolize certain nutrients, fitness, skin conditions, behavioral conditions. So these have become very common, and it's important that if you do them, or if you know somebody who has done them, that they are interpreted by someone who truly understands how these tests work. So it's important that they are reliable, valid, and accurate. So for example, these are some of the genes, again, like I said, eating behaviors. They look at how we metabolize certain macronutrients, micronutrients, food sensitivities, etc. Gut microbiome, I think you've heard enough about this morning. And then there's, of course, these are more advanced technologies in terms of we have certain genes that encode for certain, conditions. Is there anything that we can do in addition to lifestyle medicine to reduce risk of certain diseases? So CRISPR was developed, of course, and it's really revolutionized genome editing. Some of the pros, of course, it's made genome editing much simpler, faster, more accurate. However, of course, there are certain disadvantages, which I think with technological advancements, we can really try to avoid. So for example, it could edit the wrong place in a genome, but again, I think this is something that can be addressed. And finally, artificial intelligence, machine learning, and genomics, which tend to be trendy words nowadays, but have really gotten us to where we are now. Because with all the diversity in the data and the information we have, humans can only do so much in terms of, statistical analysis. So we really need to rely on machine learning and artificial intelligence to help, put all of this data together and move forward with it. So, we use machine learning techniques to identify, for example, primary types of cancers from a liquid biopsy. It can help us predict how a certain type of cancer will progress in a patient. And for example, it can help identify disease-causing genomic variants compared to those that are more benign. Okay, yes, done. Okay, thank you very much. This is also, so we've spoken about how genes can impact nutrition, but there's also more and more data how they can also impact medications and to personalize medications that are given to clients to reduce side effects and to ensure that they can tolerate them. Thank you so much. Yes.
A question, because we are late in our schedule. We're supposed to be in the. Yeah. Can you ask? Let's go for it.
And very interesting.
We need one day, Doctor. We need one day just for the genomics.
We need one huge conference with her.
And I feel so comfortable because I'm a bit overweight, not because I'm not exercising, because of my genomics.
Your chance. But then don't forget all of the environment.
Anyone have a question?
Okay, hello, my name is Joelle and I work at the Emirates Chiropractic Clinic. I have 3 questions eventually for you. The first one is about the project, the Emirati, the project you mentioned. By doing this project, I believe that we don't need any more studies. Whenever you test people, the genes of the people, it's almost like you have a huge database about almost any kind of chronic disease, or even we work on preventive nutrition more than dealing with the disease. Is it right? Or
Emirati Genome Project is actually currently still underway. Okay, so I believe about maybe 400,000 samples have been collected. The aim is to collect, to reach up to, double the amount. So if you haven't participated, please do. And it really will help in years, and I think maybe even generations to come, to help, identify what are the genes. But then with technological advancements, artificial intelligence and different methods, we will be able to extract the data. So of course, the important thing is to have access to that data, but to really be able to link it to various lifestyle markers. So, you know, I think it's something that it's still under progress, but it will be, and it's important to keep building on it. And our understanding in this field is constantly changing. So yes, we can have a subset of data, but now the next step is really what do we do with this to, you know, to reach the objectives and the goals that the Emirati Genome Project or program has been set to achieve.
Another question. Okay, who should interpret the genetic test once done? You know, we as dietitians, we refer patients to do the genetic test, but when it comes to really interpreting it, at some point, I don't know if most of the dietitians agrees, I can only interpret the dietetic part, whereas the other parts, there are things, it's beyond my knowledge and my capacity.
Yeah, and it's important to understand, you know, what's within our scope and what isn't. And there are many certification programs out there. And inshallah, as part of, you know, my work, this is something that we would like to launch in the near future. So I think it's important, you know, the same way we attend fantastic conferences like these, that we're continuing to learn. So there are a variety of online courses, you know, well-structured, well-designed, science-based that can help in interpretation. But like I said, stay tuned. And I'm happy to share my contact.
Okay, my last question is, I have a lot of, I see in my practice a lot of obesity among the children population, and then you come to see that it's genetic. Whenever you have one of the parents, it puts them at a certain risk, and if you have both parents, obese parents, so it's definitely a higher risk. So do you think I tell them that with lifestyle we can really overcome the genetic predisposition? Do you agree on this?
100%. Remember that genes load the gun and lifestyle pulls the trigger. You know, it's easy for us to blame everything on our genes. Khalas, I'm destined, you know, to have this. But again, you know, we have the power. It's what we want to do is to empower individuals to take lead and to, you know, be able to control their health. So, excuse me. Maybe there is we have genetic predisposition to so many conditions. It doesn't mean we're going to develop each and every single one, right? So it's by, you know, counseling them, setting realistic targets, and explaining, yes, if they do have that gene, but by all means, it can still be suppressed. Remember, the epigenetics is where you switch on or switch off certain genes. Okay, so we can really help, and you want to teach them lifelong habits, right? So the genes, let's say, are there, but then it's how we address those genes through practical, realistic, and lifelong changes in their lifestyle.
Thank you.
Just like to add to your question, there was a study like somatic twins from Japan, and they wanted to look at the gene effects of those somatic twins. So one of them stayed in Japan, and another one went to the United States. So you can imagine after a few years, the one in the United States, he copied the environment there and the way they're eating the Western food pattern, and he became obese, right? And the one stayed in Japan, it stays like within the normal weight as the Japanese are. So yes, the gene is there, even if they're identical, having the genotype, still the external environment is a crucial in terms of how they become. So we cannot blame it on gene. Thank you very much, and thank you to Farah and Dr. Sundus. And now we conclude the first.
I have one more question, if possible.
Maybe you can, because we are.
You are late.
We are late, very late.
Okay.
And I'm afraid that I might lose my job. So she's gonna be sit beside.
No problem.
Thank you very much.